between main stuff. Questions, queries? Any questions? Okay, so we'll complete this and I'll try to save some time for you, maybe on Wednesday, tomorrow. Tomorrow. So today we're going to talk about ear and the eye, mostly of chapter 6, 15. So please note the ear is responsible for both. And when we talk about ear, we're not just talking about the outer ear pinna, but other internal structures too. So that's why it's for hearing and balance, right? So we'll divide the ear into 3 components, right? So the outer ear, middle ear, and then the inner ear, right? So outer ear and the middle ear are just for hearing alone. The inner ear is where there'll be structures for balance as well. We'll talk a little bit. So what's the boundary between the external outer ear and the middle ear? It's the tympanic membrane, very good. It's the eardrum will be the boundary, right? So everything before that, so our ear pinna, a canal, tympanic membrane, we could generally consider as part of the outer ear, but the boundary. And then the middle ear is an air-filled cavity, which is flanked by the tympanic membrane on one side and another membrane. We'll talk about it, the oval window on the other. And bones, the smallest bones in our body, which connects these two membranes. Okay? And then the inner ear is within the bone itself. Within what bone? Temporal bone. Yeah, it's within the temporal bone itself. We have structures, and we'll describe them differently. Those have the receptors for hearing. They have the receptors for balance as well. And they are, if you didn't guess it, they are activated independent of each other. Okay? But if one does get blocked, you might have seen if you're filled up with fluid in the middle ear, your balance can be a little off. They're independently stimulated. Okay? So this image will hopefully also get us to kind of visualize the other components besides the ear pinna. What cartilage makes up the ear pinna? Elastic cartilage. And then we talked about the bony opening here. What was the opening? It leads to the canal. So the external auditory canal is this air-filled cavity. It has a tube which has hair. We'll talk about the cerulous glands as well. And then the tympanic membrane, aka eardrum, will be the boundary. So this will funnel in the sound. It will conduct the sound waves to the tympanic membrane. That will vibrate with the movements of sound. This causes these bones to vibrate as well. They'll move. And then this moves the last, is the stapes, that's the last of the bones. That moves the oval window. And that creates a vibration in the fluid within the cochlea, which looks like a snail's shell. Okay? That's for hearing. While balance is going to be a different way of stimulating. So let's talk about a few things, right? So we talked about auricle or the ear pinna. The function is to funnel in the sound. I believe our next lab is the special senses. So we'll talk about these structures. We'll look at them. We'll again review the functions of them. Okay. External acoustic meatus will lead to the external auditory canal. This will conduct the air, the sound from the outside to the tympanic membrane. Right? We do have some sebaceous gland. What's the function of sebaceous glands? It's to moisturize. What's the ceruminous glands? Mentioned this in our skin chapter. That's the one with the earwax. What does the earwax do? It tries to waterproof our ear canal. Sound will be, it conducts through water, but this way we can distinguish if we want to be waterproof, so we can travel around some other things, get stuck to our ear canal, obstructing the path. Okay? We do a pair, it is curved, which is why somebody's looking in your ear, they will actually pull it backwards and upwards before they actually pull the stethoscope in to look at the tympanic membrane. Is a nice fibrous membrane, pearly white, described as when it's nice and clean, when it's swollen, when it's inflamed, it looks red. Okay. The function of it is to transmit the sound from the external auditory canal to the ossicles. That's the common name given to the three bones in our ear. Okay? They will vibrate the bones, the ossicles. So the middle ear is also called the tympanic cavity. It's an air-filled space. Okay? There's not supposed to be any fluid there. In kids, you might have heard there's fluid in the middle ear. That's because of a different structure we'll talk about in a second. Does anyone know that structure? It has to do with the eustachian tube. We'll talk about it. The orientation is different when we were kids to what we are now. Okay? And it's the fluid in the oral cavity that goes in there. So normally, we should not have fluid in the tympanic cavity. It's air-filled. Mucous line, flank, I mentioned. Lateral, what does lateral mean? Towards the outside, so outside again, depending on each ear. They're both on the outside, so right or left, depending on where it is. But it'll be outside, then the ossicles, then the middle ear will end with the oval window, is what we're going to concentrate on. We'll talk about the round window again, but the attachment of those bones is on the oval window. Eustachian tube or pharyngotympanic tube is the one that connects the pharynx. Where's the pharynx? Yeah, the back of our throat, right? So there's usually bacteria in our oral cavity, there's bacteria in the pharynx. But the eustachian tube is to equalize pressure in the middle ear. Again, please don't just say, if this question came up, don't just say equalize pressure. It's equalize pressure in the middle ear. Okay? Because it doesn't equalize pressure in the knee, for example. Right? It equalizes pressure in the middle ear. Basically, if you have ear block, you're popping your ears, you're sending the air through the eustachian tube to get the same pressure as the outside pressure. We feel pressure can build up, right? Especially if you're congested. That's when we feel it builds up, or if you go down in an aircraft, you can also get pressurized. Right? So it equalizes pressure in the middle. It connects the tympanic cavity to the nasal tract. So when we were children, this was a lot more horizontal, right? So fluid has an easier time going in. Not really a problem. If the fluid goes in there, it's a problem if it stays there. Okay, that's why I mentioned the back of our mouth and the pharynx. There's bacteria, so bacteria staying there causes middle ear infections, right? What's the usual treatment? For the infection itself, yes, drops. But longer management?
Tubes.
Yeah, we'll put a tube, grommets or tubes. Basically, it's going to be a little tube in the tympanic membrane, so any fluid that comes in just goes out through the ear canal and not collects and waits, or it's not collecting in the tympanic membrane, cause all the infections. This had to do with the tympanic tubes again, showing both sides. They were more horizontal. As we age, it becomes more vertical, so it's not easy for the fluid to go up and still pass air very easily. So middle ear infection is otitis media. Okay, external would be externally. It's usually the canal itself. Okay? Very common in children. I mentioned. We talked about why it can be easily treated with antibiotics, but recurrent infections can cause scarring of the eardrum and can lead to hearing loss, long term as well as short term hearing loss. Right? So we don't want that. If it's recurrent infections, then one consider putting the tubes. If you're curious, the tubes will fall out after a few years, 4 or 5 years. If they don't, then we have to take it out. Again, rare. Usually they just fall out as So within the tympanic cavity, we have ossicles. Okay. Please learn the names of the bones and in what order. It's MIS in terms of order, going from the outermost lateral to the inner. That's the way sound will also travel. So it's malleus, incus, and stapes. Okay? And it's curious, I mentioned this when we were talking about joints, it's actually synovial joints that keep them in place. Okay? So they do move, they do vibrate. So the vibrations are transmitted. So the function is twofold, very important two functions. One, the one we probably guessed, is going to take the sound waves, the energy from the tympanic membrane to the oval. But another important one is also it amplifies the sound. Okay, so if someone was involved in a high-speed collision, because these are small bones with joints, they can fall. Okay, so is the sound going to travel very well? No. Okay, what we do is we make prostheses. We can actually make it from their own. We can either get the cartilage piece and just connect the two, which solves the first problem, but it doesn't really solve it. So they usually have some hearing loss. Okay, because these ossicles do amplify the sound. We're getting better. There are some prostheses which have gotten a little better these days, which are trying to mimic that and trying to amplify it. It's not perfect yet.
I just want to make sure I'm writing this down right. The malleus is the
one attached to the tympanic membrane.
Okay, thank you.
So malleus, incus, stapes. Again, remember it's going from this side, both ears, right? Tympanic membrane is the most lateral to it, and then malleus, and incus, and stapes. Any questions in the middle? Okay. So let's move into the inner ear, right? So we're going to talk here is where we're going to talk about the equilibrium part to the balance part as well as the cochlea, which is part of the ear. Here is to show how the vibrations are transmitted. So sound waves travel, funnel through our ear pinna, through the external auditory canal, will strike the tympanic membrane, causing it to vibrate. When that moves, these ossicles move, which sets off the fluid, because this last one, stapes, is attached to the oval window. It then moves the fluid inside the cochlea. I'll talk about the rounding in a second. Okay. Please note this is within bone, right? So it's not just sitting there, it's actually within the bone. These structures are present. In it, we're going to talk about the cochlea. That's for the hearing. We do have two additional groups of structures. Those are both for equilibrium, for balance, right? In terms of what they're filled with, it's very similar to CSF. Okay? Both of these are filled with similar kind of solution to CSF. They are mechanoreceptors. What are mechanoreceptors? Some sort of movement, pressure, pain, pressure, touch receptors are mechanoreceptors, baroreceptors are mechanoreceptors. These ones are also mechanoreceptors, right? They're going to sense the movement of those fluids within the structure. Both of them are mechanoreceptors, both sets rather. When I said both, I meant the hearing as well as the equilibrium component, but there are two parts to talk about. So, in terms of equilibrium, first structure is the semicircular canals. These, yes, they're not really semicircular, actually 2/3 of a circle. We have 3 of them at right angle to each other. Okay. We'll talk a little bit more about it, we'll see them more. They are for angular rotational equilibrium. So, if you were doing a cartwheel, during those movements, it's going to be the semicircular canals which send information to the brain. Where in the air is the body, the head, and the other part of the organs, the head and the bowels is. Okay. While the vestibule, the other one, that's for gravitational. These are static equilibrium, sometimes called, when we're not moving, when we're standing upside down, do a handstand, lying down. It's these structures that are being stimulated, which sends the information to the brain. Okay? So these are static or gravitational equilibrium, while these are more rotational equilibrium. Okay? Semicircular canal has receptors for rotational equilibrium, angular equilibrium, whereas the vestibules are for gravitational or static equilibrium. Here we'll talk about the two structures. For our course, we just need to recognize there are two structures. You might have heard about some crystals that's got dislodged. Some people have dizziness. It's in here, those crystals, which are the receptors themselves that get dislodged, which makes the brain unsure of what the information is. That's why they have dizziness there. We can do some movement, try and align those crystals again. Right? So these will have the receptors for positional. This is gravitational, movement, rotational. This is more gravitational. There's more movement, but in a different position, head position. Utricle and septum. So the cochlea is a spiral-shaped structure. It's going to look like a snail shell. The couple of things. So first of all, it's filled with fluid. Okay, so it's the movement of the ossicles, mainly the stapes, because that's the last one, that sets off the fluid's movement. Right, it's going to move it. If you're thinking about you can think about a syringe even if you wish, right? If you have a fluid-filled syringe, you move the plunger in and out. You're going to actually cause the fluid inside that syringe, provided you cap the other side. You're going to cause the movement of the fluid in it. Right? We have to note that the sensory receptors themselves are not spread throughout the cochlea. It's actually at the apex of the cochlea. What's the apex? Have we heard that term? Kind of the top, right? So if you had a pyramid, the top of the pyramid, the pointed area. So it's basically here that we're going to have the receptors. Those receptors, they have a couple of names. They're called spiral organ or organ of Corti. They're found at the apex. They will sense the movement of the fluid within the cochlea. Okay, they have mechanoreceptors. This is a good diagram. You can see the neurons that will form our nerves. So this is coming from the cochlea. These are coming from the vestibule. What's the cranial nerve that takes the information for both sense of hearing and equilibrium? Facial, vestibulocochlear nerve. Right. What number is it? 8. What's 10? Vagus. So here is the vestibular branch, sometimes called, sometimes, I don't know why they didn't have just name it as 13 cranial nerves, because they come from two places, they join up, but they also end up in two places. So my hypothesis, they found it later compared with the other ones, and they just didn't want to split 14 because it's right next to each other. Yeah. Anyway, so it will take the information to the brain, right? So it starts at a different spot and takes it to a different spot in our brainstem and beyond too. So, how are the information taken by, right? So we mentioned about sound waves traveling through the air, hitting the tympanic membrane, causing vibrations in the tympanic membrane, causing movement of the ossicles, which will pull the oval window which is attached to the cochlea, and that does the fluid starts to move. Okay? The movement of the fluids is what the receptors will sense. So the cells themselves look like this. They're called stereocilia cells. here they don't have the same function as regular ciliated cells. What do normal cilia do? Move stuff, right? Here they actually will stick out into the fluid, they sense the movement, and fluid moves, it's going to trigger the receptors in these cilia, the mechanoreceptors. Okay? So these are mechanoreceptors. This is a picture showing one stereocilia cell. Okay? Any questions?
Actually, not to like go back, but when you had the internal ear, what is the vestibular canal and the tympanic canal? Where does that like change? Because it looks like it's the same one in the picture.
Which one are you referring to?
The inner ear.
13.
Slide 13. Okay, thank you.
That.
So here, yeah, they have different names. They also different membranes, basilar membrane. It's just basically, it is a connection. What you can think of it, if you roll it out, it's like one continuous tube in that sense. But it's really, because it's the way it is, it's actually a double tube. For us, we don't need to differentiate. It's still part of the cochlea. It just tells which side it's oriented, closer to the vestibule or the tympanic. That's the name, nomenclature of it, but function is the same. It's just a continuation tube. Any other? Okay, so next we're going to talk a little bit about how the sound travels or where it ends up, right? So this is an image showing the cross-sectioned out cochlea. Where if you unwind it is when you see it as one tube, but it's a coiled up tube, right? So here are the receptors. It's going to take the information to the brain stem, okay, and then it goes up and goes to both sides. You may have been, I'm hoping that we have been able to distinguish which side of the room sound comes from here sound, right? This is because we do send it to both sides, but one side gets more information, right? Hence we can differentiate where it comes from. So it does send it to both sides. Okay. And where is our primary auditory cortex? The temporal lobe. Okay. And then this is to show the thalamus. Thalamus, pretty much everything. Only a couple of things that do not go, a sense of smell, for example, does not go to the thalamus. Here they've shown slivers of the different components. Here is medulla oblongata. This is to show the midbrain as a slice, just to show, again, they're trying to show three-dimensional structures in 2D. This is more of a frontal section here. These are transverse sections. So next we're going to talk about eye. You may have heard this, but a lot of our receptors, in terms of total receptors, most of them are found within our eyes. 70% of our complete body's receptors are found in the eye. So quite a lot. So hence we have a lot of structures trying to protect it. Okay. Some of these I don't have separate slides for, so we'll talk a little bit about them. Eyebrows. What's the function of eyebrows? Other than to give up some. Yeah, it's really to prevent the sweat from coming down, even some particles from coming down from top and entering the eye. What about the eyelids? Yeah, covering the anterior of our eye, preventing foreign particles from entering. Conjunctiva, we'll talk about it. It's actually a mucous membrane on the white of our eyes. You may have heard of conjunctivitis. What is conjunctivitis also known as?
Pink eye.
Pink eye, that is an inflammation of the conjunctiva. We'll talk about it. It gives an additional coating around parts of our eye. Lacrimal apparatus. Lacrimation is to
Right.
cry. Which cranial nerve innervates the lacrimal gland? Cranial nerve 7. And then extrinsic eye muscles. These are 6 eye muscles that help move the eye. Those in here, and then we'll review cranial nerves that innervate them. We'll add the detail of which muscle is innervated by which cranial nerve. Do we remember what 3 cranial nerves moves the eye? Oculomotor, trochlear, facial, abducens. So it's 3, 4, and 6 will move the eye muscle. The facial is like the tearing, and 9 is the pupils. And 7, talking about pupils are oculomotor. So conjunctiva, it's a mucous membrane. It's on the covering of the white of our eyes. White of our eyes is called the sclera. We also have conjunctiva underneath, so inside of the eyelids as well. Okay. These can swell up. So it's just a nice, smooth mucous membrane there, and it's protecting the eye. The lacrimal apparatus, it's a collection, that's why they call it apparatus. We have a lacrimal gland, we have lacrimal sac, and then we have the lacrimal duct. Lacrimal glands are located lateral side and superior to our eyes. So about here is where we have the lacrimal gland, while the lacrimal sac is located in the medial side. We looked at in the bone lab. We looked at the lacrimal, the duct's opening, right? That was going to lead to the lacrimal sac. Okay, so the lacrimal duct will lead to the lacrimal sac. This will open up into the nasal cavity. Are we tearing all the time? Right, we're producing some tears all the time. It keeps it nice and moist. We don't notice it. It's little by little. It drains in. Some of it's absorbed, some of it's swallowed. It's when we produce a lot, whether it's irritation or crying kind of reflex, that's when we notice a lot more production, and then cannot keep up with the drainage. So lacrimal gland on the lateral superior side will produce secretions. We'll talk about them in a second. Tears, which covers the anterior of the eye, will drain to the medial aspect of the eye. So they are basically lacrimal tear, lacrimation, lacrimal secretions are kind of a dilute saline solution. 1, 2, they do have some other things in it. They are part of our first line defense. They're trying to destroy any bacteria that get there, trying to prevent them from entering, right? So they do have antibodies. We do have lysozymes as well. What does it sound like? Yeah, it's talking about lysosomal enzymes. So we just produce it, secrete it into the tears itself, so that way it can break into cells that may try to enter, right? And it's the blinking that actually spreads them, right? It's a normal reflex. We actually blink a couple of times generally multiple times in a minute. Every few seconds we actually blink. This will push or will transfer the tears, okay, out of the anterior and towards the middle of face to into the nasal cavity. Okay, that's where the lacrimal sac and the lacrimal duct are. Some people have either a blockage of these ducts, which causes them to tear all the time, right? It's not because they're producing more tears necessarily, it's because the drainage is not comes out. We can try to dilate it, sometimes we do a bypass, try and collect that. Any questions? So next we're going to talk about those eye muscles, okay? There are 6. What does rectus mean? We looked at 2 recti muscles. Where did we come across rectus? Rectus abdominis and rectus femoris. What did it mean? I mentioned it. It means straight. Okay, so that means straight. So we have 4 rectus muscles. They're just named after, by their orientation, that's basically it, okay? So we have 4 rectus muscles. So we have a superior rectus, inferior rectus, medial rectus, and a lateral rectus. Okay, and then 4 recti muscles, 2 oblique muscles. What does oblique mean? Going at an angle, not in the same plane, right? We have the oblique, external oblique, internal oblique. So we have two obliques. We have a superior oblique and an inferior oblique. So I'm going to give you a little, hopefully an easy way to remember which ones are not innervated by cranial nerve number 3. Because cranial nerve number 3 innervates 4 of these 6. Okay? The formula I suggest us knowing is SO4 LR6. Okay? This kind of helps to hopefully recognize that this is talking about superior oblique. And that is by cranial nerve number 4. What do we think this one might be, Thomas? Yeah. Yeah. Lateral rectus by cranial nerve number 6. Okay, that's abducens. That's trochlea. The rest are by cranial nerve number 3, ocular motor nerve. And so we have 4 rectus muscles. They're all labeled just by orientation. 2 oblique muscles. Here's the innervation of the lateral rectus and superior oblique are the only ones that are not innervated by cranial nerve. Here's a picture trying to show that. So here you're looking at someone's left eye. Okay, you're looking at a profile view. So superior rectus. This will be lateral. It says medial rectus. That shouldn't be medial rectus. If that's the nose. That's incorrect image. So this is the lateral rectus. That's the nose. So hence that's the medial rectus. Hidden behind. That's inferior rectus. And then the obliques do a little hook kind of attachment. So this is the oblique, superior oblique, inferior oblique. So they come underneath the curve, pulls the eye in a different way to the rectus. So the opposite way, the superior oblique, the opposite direction, superior rectus, superior oblique, and the nose. Next, we're going to talk about the lens. So we're going inside. We'll talk about the layers in a second. We have 3 layers. That's why I kind of skipped over the sclera and the cornea. We'll talk about that in a second. These are other structures. This is within the eye itself, right? What's the shape of the lens? Is it flat? No, it's convex. What does it mean, convex? Biconvex. Which way? Outwards, right? Convex is curved outward, concave is curved inwards. Especially younger individuals, they have glasses, they are usually wearing concave glasses. You can see with the edges, they'll be wider compared to the middle. Okay? But our lens is a biconvex lens. Okay? It's transparent, it's flexible, and it's avascular. Okay? So it doesn't have its own blood supply. So we'll talk about how it consists of nourishment. Okay? The function is to change shape to help focus the image on the retina. This is not the only thing that actually bends the light. This is actually less bending compared to our cornea that actually refract light a lot more than our lens. Okay? As we age, it does become more dense, it becomes less elastic, and then we develop cataracts, right? So cataracts, the modification of the lens. What's the treatment? Surgery. It's a pretty cool surgery actually. He says, I know this class usually likes this description. It's pretty cool. So they put a little incision underneath the where the sclera and the cornea meet. We put a little, it's like a little probe, it's almost like a little blender. Okay. Put that in, it blends the lens, and then it suctions it out. So we don't have to remove the whole lens, otherwise we have to put a very large incision, right? Because this lens is not moved, it's not very pliable, it's very solid. Okay? If we have a chance, when we do our special senses, we can dissect the cow's eye. You're going to see they have cataract, those eyes. Those lenses are, they look very, they are hardened, they look like cataract eyes. So you can't just take it out without, unless you put a big incision. So we just put a little one, put this little probe in, it's called phagocoagulation. We blend it, suction it out, and then we put somebody else's lens? No. We put similar to contact lens, the only difference is it's got two springs. So we fold the lens, we put it in, we let it go, the two springs will hold it in place. Okay? That's pretty cool. And it's done within really the surgery itself is very short, it's like 15 minutes, it's the prep and so on. But then they still go in morning, couple of hours, 2-3 hours, and they actually usually can go home afterwards. They just can't drive straight away to the drops, but we just go home straight away. That's pretty cool. Some things do increase the formation of cataracts. Okay, heavy smokers, we see the cataracts developing early, even in the 40s. Somebody who has hypertension, we see them developing earlier. People who are working outdoors, if they don't wear sunglasses so often, they are going to be exposed. They will develop cataracts early, right? So in terms of black and white, what's the difference in terms of physics difference? Does anyone know? The color white, the color black? Yeah. Black actually retains all of the rays of light. It does not bounce back. While the opposite happens in white, it bounces all of the rays back. Okay? So if there's a lot of exposure to sun, what's happening? Kind of an adaptation, we're trying to reflect that sunlight. Okay, so they develop the cataract early, which is becoming cloudifying of the lens itself. It's not the iris, it's not the pupil, it's the lens that becomes that, right? So the light has to pass through, we'll talk about it in a little bit, passes through the cornea, it passes through the anterior cavity, then passes through the lens, then the vitreous humor, and then into the retina, right? So this is just behind our iris, the pupil is the opening of the iris. So it's just that's what we see as white. That's qualification of the lens. So it can happen in the 40s, 50s. Usually it's in the 60s, 70s is when we see it, unless some other risk factors, diabetes, heavy smoking, exposure to sunlight. Next, now we're going to talk about the layers of the eye. Okay, so we have three layers of the eye. Outer layer is the fibrous layer. Middle layer is the vascular layer. We'll talk about different parts of each. And then the innermost layer, that's our retina, the neural layer. Okay. The internal cavity of our eye is filled with fluid. The fluid, another name is humor. Okay, there's no U there, not the second U there. This is still talking about liquids. We'll talk about it. One is more like water liquid, the other one's a gelatinous liquid. Okay, I'll talk about it in a second. So the lens basically separates the internal cavity of the eye into anterior cavity and a posterior cavity. So behind the lens is posterior cavity, in front of the lens is anterior cavity. And they're filled with different solutions, different treatments. So here is a side, it's basically a sagittal view, but you're looking at sagittal section, you're looking at a profile view. So this will be the first part, that's the cornea, and light will pass through. We'll talk about these two cavities. Here's our lens. Anterior to the lens is anterior cavity. That's filled with aqueous humor, a little bit more solution, more like fluid, more liquidy fluid. And then the posterior cavity behind the lens all the way to the retina is posterior cavity filled by a gelatinous humor, gelatinous solution called vitreous. And then we'll talk about the layers in a second, so I'm just going to introduce it here. The outermost and the fibrous layer is sclera and the cornea. So sclera is the white of our eyes, while the cornea is the clear part way in front. That's where the light passes through. In terms of the vascular layer, the middle layer, there are three parts. There is the choroid. So the blue, the red is the middle part, and then the yellow is the innermost here. The choroid, then in front, you may have heard about it, maybe not, ciliary body. Hopefully you've heard about the other one. The iris, they're part of the middle layer. Okay? And then the innermost layer is the retina, also called the neural layer of the eye. Okay, we'll go back. So let's talk about the chambers, put them into words. Internal cavity, the anterior cavity is filled with aqueous fluid. This is very similar to our CSF, right? Where is CSF produced? Within the ventricles with what structure? No, that connects third to fourth. Yeah, that's very correct. The choroid plexus, which is blood vessels, and the ependymal cells, right? They'll produce it. Do they just stay in the ventricles? Where else can we find CSF again? Subarachnoid space. And what about the spinal cord? Around the subarachnoid space, but within the spinal cord? The central canal also contains CSF, right? It's produced and then drains, right? So we mentioned about the arachnoid villi, it goes back into blood circulation. Similarly, aqueous humor is continuously produced and drained. Okay? It should be matched, and then we'll talk what happens if it's not, if it's mismatched. So it's continuously produced from the ciliary. It's part of the ciliary body. If you said ciliary body, please answer okay with that. Ciliary process, and then it's going to be the ligaments part of it, the ciliary muscle. That's what we're going to talk about. So it fills up the anterior chamber or anterior segment, and then it will drain in a structure called canal of Schlemm. It's at the boundary where the cornea and the sclera meet. It is a continuous process. It will provide nourishment, nutrients, and oxygen to our lens. Why should it provide nourishment to the lens? It's avascular. we haven't talked about this, but we will mention it shortly, even the cornea is avascular, which is why we can have corneal transplant and not need immunosuppressants. Okay? Otherwise, with the kidney transplant, liver transplant, you will need immunosuppressants. A corneal transplant does not. It's not a surprise to it. Okay? So it depends on its aqueous humor for its supply needs as well as removal of waste. Okay? Both of these are avascular. Okay. So let's talk about what happens if we have too overproduction or under drainage. You may have heard of glaucoma. That's because this fluid collects, right? So you would have heard it as pressure in the eye. Okay? Let me go back to this image. So this fluid is found in this space. Okay? It's produced by these structures here, part of the ciliary body. Ciliary process is not shown, it's more microscopic. It's going to fill up this area and then it's going to drain through this port. Okay? If it's under draining or overproduction, we'll have this fluid collecting and push us forward, but also push us backwards. Right? When it pushes backwards, the lens is going to get pushed back, and it's going to push back on the vitreous humor, which also pushes on the retina. Right? What will happen if we keep applying pressure on our skin? And if it's excess pressure, it's going to cause some trauma there. Okay, that's what happens here too. Glaucoma, we don't want to leave it untreated because you're going to lose the retina cells and it's squished. Okay, so it's glaucoma. So first, initially we treat it with eye drops. We're trying to increase the drainage, right? Later on we could treat, or if it's an acute. So if you have two, I would say, a definite ophthalmic emergencies. If you want to see an ophthalmologist, not an optician, not an optometrist, but you want to see an ophthalmologist, which is a medical doctor. The training for that is you have pressure in your eye that comes out suddenly. You want to see them, and if you have like a curtain effect, if somebody present with a curtain, you want to go soon. I'll talk about what that is. That's retinal detachment. You want to get that attached soon as possible. Well, yeah, glaucoma, we have acute glaucoma, and then most people suffer with chronic glaucoma, right? Where it's just eye drop, but it's acute, we have to do laser to try and quickly relieve that pressure, otherwise there will be long-term loss in sight. Of course, trauma, any foreign body, you want to go ashore. These two. So posterior cavity or posterior segment is filled by something called vitreous humor. This is gelatinous. If we do, hopefully if we do dissect the cow's eye, you're going to see the vitreous humor. It's still gelatinous aqueous humor. You can differentiate from the rest of the solution that's sometimes put to preserve it, but we generally do see the gelatinous nature of vitreous humor. Vitreous humor is lifelong. It's produced once and that's it. Cannot be replaced. Okay. So there are some functions to this vitreous humor besides just filling up a space. Okay. They kind of gently hold the lens in place, but very importantly, they hold the two layers of the retina in place. Okay. If there's not enough vitreous humor, the retina can come off the other part, which is basically retinal detachment. Right. So it has to last a lifetime. It holds the two layers of the retina together. Just put gentle pressure on it. Hold it like two pieces of paper. There's a gentle pressure. You don't want too much pressure either. We want gentle pressure on it. So let's talk about the layers. Right? Actually, let's take a break there and then we can talk about 3 layers.